back with our next class. In today's class, we'll start with bacterial reproduction. Alright? So without any delay, let's start. Bacterial reproduction can be of two types. Asexual reproduction and sexual reproduction. Now, asexual reproduction, you know, is a process in which there will be no genetic recombination involved and the daughter cells produced will be exact clones to the parental cell. So in bacteria as well, asexual mode of reproduction would be occurring mostly, and during favorable condition, most bacteria would undergo binary fission. Alright, that's the most common type of reproduction observed in bacteria. But then also under unfavorable condition, there can be other processes of reproduction or perennation that can be observed in a bacteria like endospore formation. Alright, so we will be discussing under asexual mode binary fission and endospore formation. Now, regarding sexual reproduction in bacteria, see, sexual reproduction in bacteria is not that typical sexual reproduction where the gametes will be formed and fused and then biparental or any such thing. Okay. Sexual reproduction in bacteria simply means the transfer of genetic material from one bacteria to another that will give rise to genetic recombination. So that's why we also consider the atypical sexual reproduction in bacteria to be parasexuality as well. Under sexual reproduction in bacteria, we'll be mainly discussing three modes, that is transformation, conjugation and transduction. Alright, so let us start with asexual mode. So now, let us start with the asexual mode of reproduction. As I've already told you, the most common mode of reproduction in bacteria is binary fission. And during favorable condition, a bacteria would typically undergo binary fission every 20 minutes. Alright, so in that case, let us start or let us see how binary fission would occur in a bacteria. First, suppose this is the parental cell. Alright, now the parental cell, when it starts to divide, first thing that would happen in the cell is replication of the DNA. Now, as you know already, the bacterial DNA is double-stranded and circular in nature. It undergoes a method of replication which is called theta model of replication. Because while the DNA will replicate, at one point the parental DNA and the newly formed daughter DNA both will look like the Greek letter theta. That's why we call it theta model of replication. Alright. Now, as you can see, the newly formed DNA or the daughter chromosome also would get surrounded or attached by a newly formed mesosome. Alright. And then a membrane will start to develop between the two mesosomes. Alright, a membrane will start developing between the two mesosomes, and that's why the mesosome will start moving apart. Okay, this mesosome will start moving towards this direction, this one will start moving towards the opposite direction. That's why both the chromosomes would be separated from one another and they will reach opposite poles of the cell. So, karyokinesis will complete like this, or nuclear division is complete. Alright, now cytokinesis or cytoplasmic division has to occur. See, for cytoplasmic division, exactly at the center of the cytoplasm transversely, a centripetal movement starts. Okay, that means the plasma membrane starts to form an inward constriction that starts moving towards the center. So we call it centripetal movement. So this is known as furrow. So furrow formation occurs here. Alright, as the furrow starts to deepen, the cytoplasm, as you can see here, have separated. So there are the protoplasm here between the two daughter cells have equally divided and have been separated. Alright. So here do remember the karyokinesis as you have seen or the cytoplasmic division. Both the division methods that you are seeing here, there is no spindle formation. So in bacterial binary fission, as there is no spindle formation occurring, so we can call such type of cell division to be amitosis as well. Alright. So see here, as the cytokinesis completes, now each of the cytoplasm will start secreting cell wall towards this particular region. So this cytoplasm will form a cell wall here, and this cytoplasm will also secrete a cell wall here. So that's why at a particular time there will be a cross wall that will be formed between the two daughter cells. So here we can write cross wall formation. So after cross wall formation, finally you can see that, or you can, what would happen is that there will be a turgor that will develop in this cell, and due to that the two daughter cells will separate. So daughter cells separate. Alright, so this is the most common mode of asexual reproduction that is seen in bacteria during favorable condition, and that is binary fission. So because in a typical bacteria, binary fission would be occurring every 20 minutes. That's why from a single parental cell within 24 hours, okay, within 24 hours, some 4.7 into 10 to the power 21 cells or descendants can be formed. Alright, that's a very important thing for you to remember. Alright, so that was regarding binary fission. Now the next thing we'll discuss is endospore formation. That is also a asexual mode of reproduction in bacteria, but then would be only occurring during unfavorable condition. So let's see how it occurs. Now let us discuss endospore formation. Alright. Now unlike binary fission, endospore formation is going to only occur in bacteria during unfavorable condition. Alright. So it is more like a perennation process in bacteria. That is a process that helps the organism to survive unfavorable condition. Alright. Also in this process, one parental cell will give rise to only one daughter cell. Alright, so it's more of a perennation process rather than reproduction process. Fine. Now, if I'm telling that endospore formation only occurs during unfavorable conditions, so what are those unfavorable conditions in which a bacteria gets induced to form an endospore? Now, there can be various types of factors responsible, like the most important is lack of food or lack of nutrients in the surrounding of the bacteria. Apart from that, there can be toxic accumulations in the surrounding or else sudden heat shock. Anything can induce a bacteria for endospore formation. Now let us see how endospore will finally develop. Alright. See, suppose this is a bacterial cell or parental cell. Alright. During endospore formation, whenever the bacterial cell or the parental cell is undergoing some or the other unfavorable condition, a part of the cytoplasm of the cell containing the nucleoid will start to get dehydrated. Alright, and then will also start getting surrounded by a membrane, okay, similar to plasma membrane. So this part that has separated from the rest of the cytoplasm, this is now going to be called as endospore primordia. Alright. Now, the rest of the cytoplasm will keep secreting certain wall layers surrounding the primordia, so that surrounding the primordia many wall layers will start forming, giving rise to a well-developed endospore. So this well developed endospore. And it's going to be a very large structure containing so many wall layers, as you can see, like the closest one, which is the most internal coat of wall, we call it the core wall. Core wall, alright? After the core wall, next layer is called cortex. The cortex is very thick and consists of peptidoglycan and most importantly an anticoagulant called calcium dipicolinic acid complex. Okay, it consists of calcium dipicolinic acid complex. This gives the endospore a kind of a very important protective layer, and it will be saving the endospore from desiccation. It will not allow the endospore to dry up. Next layer is going to be called as spore coat. Spore coat. Alright? And finally, as you can see, this thickest layer which contains air, this is known as exosporium. So we first have a core wall, then we have a cortex containing, that is made up of peptidoglycan and calcium and dipicolinic acid complex. Then we have spore coat and finally exosporium. The dehydration in the cytoplasm makes the exosporium very light so that it can easily float in air. Okay. Next, here you can see I have made the diagram of certain bacterial cells with well developed endospore. So the endospore being such a massive structure, whenever it will be formed inside a bacterial cell, that part of the bacterial cell will swell up. Alright. So these red colored marked circles, they are all endospores. They have swollen up the parental cell. So parental cell swells up at the site of endospore formation. Alright. Finally, because it is constantly pressurizing the cell wall of the parental cell, the endospore will rupture the cell wall and it will be released. So, endospore is released by rupture of the parental cell wall. So as the endospore is released, it can easily withstand a lot of harsh condition in its surroundings because it is a highly resistant spore. It can easily withstand high temperature, say temperature of plus minus 100 degrees. It can easily withstand desiccation. So it can withstand majority of the harsh conditions. And finally, when favorable condition will return, the endospore walls will break down and the primordia will be released. This is the primordia. Only this section of the bacterial cell containing the nucleoid, only this will be released. So primordia released. And as it is released, finally, this primordia will eventually commence into a daughter cell. So a single parental cell will eventually, during unfavorable condition, form an endospore. Due to endospore formation, the parental cell will swell up at the site of the development of endospore. Eventually, the parental cell wall will rupture, due to which the endospore will be released. And when favorable condition will return, the endospore primordia will come out by rupture of the endospore cell walls. Okay. And then finally, this primordia will act as a daughter cell and will commence, or it will start growing as a new cell. Okay. Because of that, we call endospore formation to be a reproduction process, because at the end of the process, when favorable condition returns, a new daughter cell is going to be formed. But then main purpose for endospore formation was not reproduction, rather it was perennation. Is that clear? So now, asexual mode of reproduction being discussed, we'll move on to the next part, that is sexual reproduction. Alright, and that we'll do in the next class.